Okay, so welcome to everybody and welcome to those of you who are coming in now. Welcome to today's bootcamp webinar. I'm Rebecca Steele, I'm your host for this session today on drugging the RNA world. Today, our speakers are Stefan Lohmer, our CEO and CSO, and Scarabottolo, our Director of Discovery Services. This presentation today should last around about 45 minutes. And then there'll be some time for questions afterwards. So if you have any questions, please submit them in the Q&A section, and then our speakers will do their best to answer them at the end of this presentation. So now I'll hand you over to Stefan and Leah and enjoy the webinar.
Thanks, Rebecca, for the nice introduction. So good morning, good afternoon, good evening, wherever you're on the planet are. Welcome to this seminar with the title Drugging the RNA World. So before coming to the RNA world, just two slides introducing you to Axxam for those who are not really familiar with Axxam itself. So putting in a nutshell, our activities is as a key expertise is identification and the generation of bioactive molecules applying high throughput screening. So therefore we have all the different kind of modules here in place. Right now, we're a little bit more, even more than 100, so roughly we're 160 employees in Axxam so far. We are working on 3 different kind of locations. So the headquarters here where we're talking also today is here in Milano, in Bresso, where most of the people are. Then we have a small chemistry team down in south in Naples. And then recently the Germany, the Axxam GmbH is based in Constance on the lovely lake of Constance. Offices we have running over in Cambridge, Massachusetts, and also in the Cobis in Copenhagen. We started our business more than 20 years ago as a spin-off from the Bayer Group and from the business segment. We're actively in all the different kinds of in the field of life science, ranging from pharma, biotech, agrochemical over to food, flavor, and fragrances and cosmetics as well. So we are organized, what you can see in the next slide, and this kind of different kind of modules, where in the heart, in the center here is high throughput screening, where we do most of our activities either in a 384 well or 1536 well format with all these different kind of readout systems ranging from fluorescence, luminescence over to radiometric high content, but even including, which you will see later also on high throughput screening using TechMan, that means RNA detection system, and also all different kinds of electrophysiology. And all this kind of screening are fed and made on purpose, fit for purpose assays, which are ranging from cell-based biochemical assays, but of course using also phenotypic assays, and then the newest generation also of iPSC derived cells, using also here gene editing or also optogenetics. So that is a major flow here in Axxam, meaning assay development coming over then in high throughput screening and using then different kind of compound collection, either ourselves or those one from our partners and clients. But now coming after this short introduction to the topic of today, so drugging the RNA world. So as most of you which are in the industry for several years, also you realize this kind of trends here that there are new approaches necessary. So the classical biotech pharma industry in the 90s and 2000, so they already picked all this kind of low-hanging fruits on this wonderful apple tree, but still many targets remain, and these are the more the high-hanging fruits where the industry is struggling right now in the 2020s. So most of those interesting targets are so-called undruggable, or they have a rather higher challenging therapeutics index. But let's say with the right ladder here, with an enabling technologies, you can also do the higher hanging fruits. And one of those higher hanging fruits is, of course, in this world, drugging also RNA. So, and this is something normally what we learn in textbooks or in older textbooks. Of course, you have here your very nice double-stranded DNA, which is transcribed into RNA, which is translated into protein. But this central dogma in molecular biology, of course, has created some kind of deviation, but also even further insights right now, which were coming out by the recent, let's say, last 15 years of deep RNA sequencing, where many other RNAs are as well also were discovered. But before coming to the new ones, so the classical ones, of course, which are indicated here in green, which are important to make the protein, of course, is the trivial mRNA. Then, of course, you need the tRNA and the ribosomal RNA. And when you put all these RNAs, those three greens together making a protein, then normally 98% of your overall RNA content in a cell is already covered. But then more recently, so you have then more this miRNAs and the long non-coding RNAs, but also cRNA, pRNA, and so on. So most of them, particularly those ones which are here indicated in blue, they have a very important regulatory function, which are involved in controlling translation or transcription on both sides. And therefore, there is a whole plethora of new molecules which are based on mRNA, based on RNA, which are interfering with the overall process here as well. And coming to this in a second, but of course, most of the people, let's say the 20 years, they regarded all those kind of RNAs they discovered as garbage. But now we know that they're very important in a regulatory role over as well. So what you can see or what you can recognize when you go in the ENCODE, so in the ENCODE, you will discover right now that 70 to 90% of the DNA is transcribed to RNA. So previous textbook knowledge was just telling you that maybe even only 5% of the DNA is transcribed in RNA, but over the last 15 years, there was really a revolution ongoing. So that now we know, even in some articles, you can even read that 90% of the DNA is transcribed into RNA, but only 3% of those transcripts, which we know already, are coding for protein. So all of the rest of what is transcribed is non-coding RNA. So therefore, let's say, based on those kind of discovery which we were made in the last 10 to 15 years, we're changing a little bit this protein-only mindset in drug development, also to discover or to recognize also RNA as a potential therapeutic target. I think what really sets the trail or the train completely to switch over also, let's say, to consider RNA also as a good target for small molecules was the discovery of Risdiplam, which is now also on the market. And this is a small molecule, which is a splicing modulator. And I come to this in a second. So I think this was really one of the particular breakthroughs which recognized also RNA as a good or excellent target or available target also for small molecule. So therefore, what is also interesting when you look closer to the structure of RNA, so they have, many of them have a stable tertiary structure, and also in some cases they have even a very interesting quaternary structure. So they're coming always closer, even like to protein structure, so that you have really an interesting 3D structure, which makes them really amenable, suitable also for the interference with small molecules. So that gives us, of course, by those kind of folding, a big opportunity or at least a new opportunity to selective targeting these RNAs with small molecules, because by the end of the day, also the RNA is nothing else based on the chemical blocks there, like DNA, let's say hydrogen donors with charges, without charges. So it's not this quite different also from, let's say, amino acids, or let's say from proteins. So, but what is also interesting or important to know, so that small molecules are not drugging RNA is not a complete new story, because what you see here on that nice table here are antibiotics, and all those antibiotics which are very well known, is this streptomycin or neomycin, or is this tetracycline, they are all interfering, they do the interaction by binding of ribosomal RNA, either the 16S ribosomal subunit or the 23 ribosomal. So of course, this not are all, let's say like this one here, all bona fide, what I call small molecules. There are rather already some larger molecules, but some of them, they still have really this nice character of a small molecule. Therefore, it's not a completely new story that small molecules can interfere with RNA with a therapeutic index. So therefore, there are many, let's say, already examples on the market where small molecules are interfering very specific with RNA. So that sometimes people are forgetting, but we're using those kind of antibiotics since 30, 40 years and even longer, but they're really bona fide RNA binders. So, and they're doing this interaction directly on the RNA. So this is just pushing back or going back to the history, just setting the ground. It's not really totally novel or revolutionary there. Okay, so on that table, again, even more examples. So before you saw these antibiotics, what you can see now are some additional interesting molecules, and all of those molecules which are mentioned on that table here either are already on the market or in preclinical, clinical, or already on the market as well. What you can see here, so this ribosomal RNA interference here is going on directly on the modifying the tRNA direct interaction in Duchenne disease, this kind of small molecule called atolurane. Then you have here a plethora of viral RNA interfering small molecules, either interfering here with the HEV TAR protein or RNA directly, or with the HEV, with the famous iris element here, but even SARS is here involved on this COVID pseudoknot. So there have also molecules, but also the influenza A promoter RNA with this DPQ. So there are already a lot of those examples where directly small molecules are interfering, binding directly to RNA. Then what you know also from textbooks already, that this famous kind of riboswitches, and then you have, these are all the native or natural molecules which we know, which are really coming in nature, and those are those ribosyl A, B, C, which are derivatives, and they're made artificial on the synthetic way, doing the same kind of job, making this very specific riboswitches. So then what I mentioned already before, which was really, let's say, the big breakthrough in the last year regarding small molecules in RNA interaction. So splicing modulators or modifiers, meaning working on the precursor mRNA. So there you have those two examples where now Risdiplam from Roche is on the market. The Branaplam from Novartis, they do it now in a different indication, so they will use it in Huntington disease, but both of them are splicing modulator, which are quite important right now to modify splicing on introns exon border so that you can really nicely modify this interaction, which is particularly important when you have also some kind of loss or gain of function, genetic background diseases. Last not least, where I think also some of you are also familiar with this oncogenic microRNAs, particular on the microRNA 210, but also 96. So this 96, of course, this is not maybe really what we would call a bona fide small molecule. This lower already is more in this range of a small molecule. But anyway, they show also a very specific interaction directly with RNA. So this is really setting the ground so that you have different, let's say, all the different ways where we can think about a ribosomal RNA, but also on mRNA or microRNA, you can find small molecules which are binding, interacting very specifically with those RNA changes in the organism, and set really the train to find really therapeutics interacting then directly at that level, which maybe was previously more called as undruggable, but we learn right now that this is also possible over here. So this was this kind of introduction from my side regarding this one. This is maybe the newest wave, and then we have to see how this is building in the future. I think most of you who are listening, you know the PROTAC story. So where you have really the bispecific, which are bringing E3 ligase close to your protein of interest for degradation. The same is also now playing out, and this is still in the phase to see if this is a general mechanism. But here you have your RNA of interest, where this is specifically degraded, you know, of course, our specific, which is really harboring here, bringing close specific RNAs, which is already in the cell. So these are going on the first works, and this is also several publications you can find right now. This is called, according to the PROTAC, it's called the RiboTag, which is then also a nice mechanism, particularly when you have a gain of function mutation or overactivation by your genes, so where you can make a specific degradation already at the mRNA level. You don't need to wait until the protein is made, but you can eliminate already very specifically at the mRNA level by that kind of course, you need a small molecule, okay, it's a little bit larger, which has a binding activity to a recruiting activity to the RNAs and a very selective binding motif on your RNA of interest. So there the first papers are coming out on that kind of technology. Of course, it's still what I would say in the embryonic phase, like a prototype was maybe 10 years ago, but it has also certain promises also here to play in the future an important role. So this is a kind of an introduction to drugging the RNA, and Leah will show you now what we're doing here in Axxam regarding those kind of assays, particularly addressing RNA, makes those RNA assays ready for high throughput screening. So I hand over now to Leah.
Okay, so hello everybody. Thank you, Stefan, for this very nice introduction to the RNA world. So in my part of the seminar, I will talk to you on the RNA focus assay that we developed at Axxam. And I would like to start by showing this pie chart. We still illustrate the interest and the potentiality of druggable and coding and non-coding RNA as a druggable target. So since, as already Stefan explained, our core competencies are focused on the early stage of drug discovery, we decided during the last year to invest in order to generate a sort of platform for the development of different assays which tackle RNA, with the final scope to use this kind of assay for high throughput screening for identify molecules, small molecule, which can interfere with the different RNA classes. So here I have listed all the kind of assay for which we have developed, and specifically we develop assay for RNA alternative splicing, but also we have experience in RNA silencing by means of siRNA or SAH RNA. We generate assay in order to identify molecule able to interfere with RNA protein interaction, as well as messenger RNA transcription modulator. And we also develop assay for RNase enzyme and RNA replication. Based on the type of target and on the final goal, we can, of course, develop different kind of assay. So first of all, for the cell-based assay, we can use a different kind of cell line. So iPS-derived cell, or tissue-specific cells, and also in some cases recombinant cell line. We generate also cell-free assay, and in this case, beside the development and optimization of the assay, we also take care of the protein and enzyme expression and purification. All the assays are developed and optimized in miniaturized format, so in the 384 microtitle plate format. We set up functional, phenotypic, and plate-based assay using different readout systems, which include luminescence, fluorescence, tear threat, fluorescent polarization, and gene expression by Taqman-based analysis. In all the cases, we set up the negative control or so-called interference assay in order to fish out and to exclude all hits which are just toxic or unspecific. And all these assays need to achieve very stringent quality criteria before moving to the assay optimization for full automated high throughput screening. So in my presentation, I will show you some experimental approach that we use to develop this kind of assay, and also some case studies. So starting from targeting messenger RNA splicing with small molecules. So here I just want to recall that more than 90% of the human protein coding genes undergo alternative splicing. And alternative splicing is really complex, and so it includes mutually exclusive exons, so the inclusion of alternative first or last exon, and also alternative splicing sign and intron retection. So as you can see, very complex. And it has been demonstrated that the brain shows the highest amount of alternative splicing. And why it is so, I would say, meaningful to target the alternative splicing is that because in this case it is a very favorable approach, since we touch the early step of the gene expression and we are not interfering at the genome level, so without alteration of the genome, and so reducing potential side effects. As very well described by Stefan, there is a proof of principle that small molecule can indeed be able to modulate such a kind of effect, so this alternative splicing. And in addition, increasing number of neurodegenerative pathologies which are related to defective alternative splicing led to increasing interest in its splicing assay and also in the development of focus libraries. So here I have sum up the different type of assay that we use for analyzing and for the screening of alternative splicing modulator. So first of all, the Tachman-based assay. So this we can call the gold standard technique to identify and to quantify the variant, splice variant. So in this case, we can use and we can detect in a single way, and so in a single cell lysate, different splice variant together with the housekeeping gene. So there is a possibility of multiplexing, and so again, in the same way, having the multiple detection. And this kind of approach is, of course, ideal for primary cell and post-mitotic cells, such as iPS-derived cells. In any case, this assay is suitable for primary screening, and I will show you later that we have indeed integrated in an high-throughput screening station this kind of devices, and it's also particularly useful as a secondary orthogonal assay for hit validation. Then we have also additional approaches which rely on the use of the reporter gene. So in this case, we call this approach as a mini gene reporter assay. So in this case, we have to generate a construct, a plasmid, which expresses luciferase in frame of a specific exon, which is typically included, for example, in the aberrant variant. So this is just to exemplify a construct in which you see that in the presence of a splicing variant, we have the exclusion of the exon 3, and so we have the skipping of the stop codon and the inclusion of exon 4, and so the expression of luciferase. So in the presence of the aberrant splice variant, we will have expression of luciferase, and so we record a luminescent signal. While in the presence of the normal variant, the luciferase won't be expressed, and so we will have dark. This kind of assay need to use a recombinant cell line, but is particularly useful for primary screening when a very large compound collection needs to be used. And so in this case, what we are going to record is a luminescent signal, and this can be really done also in a cheaper way. But what has to be underlined that in, I would say, most of the cases, then we recommend to use as a secondary assay the Tachman assay, so in order to validate the hit which are identified by the use of this system. Then we develop other assay by the use still of the luciferase as a reporter system, but in an endogenous cell line. So in this case, what we did is that we tag with a hybrid tag endogenously the different isoform, and so the normal variant and the alternative splice variant. In this case, we have to tag the two variants into separate cell lines. And it's very well known that in order to elicit the luciferase activity and so emission of luminescence, we have to complement the IBIT by a transient transfection of large bits. And so in this case, we create the nanoluciferase and so the emission of a luminescent signal. And so in this case, upon the activation of either the two isoforms, so the normal variant or the alternative splice variant, we have the emission of luciferase. So the way to engineer this cell line is typically done by the use of CRISPR-Cas9 technology. Also, this kind of approach is particularly suitable for high-throughput screening, but also in this case is very well associated and combined to the Tachman assay as a secondary screening for hit validation. This is a workflow that I wanted to report you for the use of the messenger RNA analysis by Tachman assay using our machine and also our high-throughput screening station. So in this case, we see the cell in 384 plate, and then we treat the cell with the compound. Then, of course, we proceed with cell washing step, and then we lysate the cell, and we dilute and transfer the lysate to the plate, which are then added with the Tachman reaction mix. And then we process the reaction, the qPCR reaction, and then we got the fluorescence result. It's important to underline that for most of the cases, we optimize a one-step reaction, and so without the need of RNA purification step. This reduces a lot the time, but also reduces the variability. Of course, if needed, we can also apply the two-step reaction mix. In a single lysate, and so in a single well, we can detect and we can quantify the expression of different variants. And this is possible by using different probes, which of course carry different fluorophore. And so in this case, we can simultaneously quantify the different splicing variant together with the skipping gene, which is required for normalization. I have reported some case study in which we run isotope screening using this approach, and so a Tachman-based screening. So in this case, we screen a compound collection of 20,000 compounds in order to identify compounds which were either able to inhibit the pathological splice variant or to induce the normal, the healthy splice variant. And this was run using this simultaneous quantification, so multiplexing, and the analysis was performed by the use of gene data screener. So this is a specific screener for isotope screening, which a specific software which we have in place for all our HTS, and we recently implemented also for the Tachman screen. And we had a very successful screening. So in the end, we could identify molecule able to inhibit the pathological variant or to activate the healthy variant. But also we were quite lucky because we could identify some molecule which were able to behave in the two conditions, in the two way. And so here I just wanted to report you an example of the result of one of these very interesting hit, in which in the presence of increasing concentration of the compound, we have no influence on the housekeeping gene, while we have the reduction on the expression of the aberrant splice variant and an increase of the expression of the normal splice variant. This is another case study in which we run a screening by the use of our compound collection that I will describe later on, which is composed by 2,200 compounds, which are specifically focusing splicing modification. And so in this case, you see here the schematic representation in which the normal splice variant does contain the 3 exons, while the aberrant splice variant skip the 2 final exons and vice versa, include a second exon. In this case, we needed to validate the assay, so we had no available reference compound to validate the assay. And so we proceed in this way. We focus on a specific splice factor, which is responsible for the activation of the normal splice variant. And so here you can see that in normal condition, we have a high level of expression of our splice factor, and consequently a very high level of expression of the normal variant. But when we silence this splice factor by the use of sirna, we have the other way around, a very strong increase of the aberrant splice variant and the reduction of the normal variant. And we optimize this assay, I would say, using this condition in order to run the screen in the most proper condition to identify the final hits. But in parallel to the use of the Tachman-based screening, we also develop another assay by approaching the normal variant, by tagging the normal variant by the use of IBIT. And so in this case, what we did is that we could detect the expression of the normal variant as increase of luminescence. Also in this case, we wanted to validate our assay by silencing this splice factor. And here you can see that that in the normal condition, we have a very high level of luciferase, which is corresponding to a level of normal variant expression, while upon the silencing of this splice factor, we are in the presence, so we obtain a very significant reduction of luciferase. And by running the screening also using such a kind of approach, also in this case, we could identify the very interesting hit and also hit which behave as activator of the normal variant and repressor of the aberrant variant. The last example for the alternative splice variant is related to the nanoluciferase mini gene, which I mentioned at the beginning. And so in this case, the healthy condition is indeed a balance of the splice and the unspliced variant. But in the disease, we have a very strong increase of the unspliced variant. So what we did is we create a construct in which we put the nanoleukin frame of this exon 3, which is indeed expressed only when we are in the presence of the unsplicing, and so of an alternative aberrant splicing. And so, as we observe, is that in the normal condition, we have a low level of luciferase expression, so low level of luminescence, while when we skip the exon 2, and so we are in the presence of the disease, we will obtain a very high expression of the luciferase, and so of luminescent signal. Just to give you some information about this collection of splicing modifier small molecule. So we collected this compound collection starting by analyzing all the compounds which are available on the market from different providers, but we focus only on the most recently synthesized compound. That we applied some sequence filter, so the smart string approach, using key feature of the active compound, which are demonstrated to be active as a modulator of this alternative splicing. And then we apply a filter for physicochemical properties and also Payne's filter in order to remove frequent hitter. Then we focus on scaffold selection, which are considered privileged scaffold for RNA interaction. And so we came up with a total number of almost 2,200 drug-like compounds, which are selected for I2PO screening for drug discovery. And so interesting to note that almost all these compounds fall in this square of the so-called drug-like properties, and most of them fall in this lead-like square, in which we have a molecular weight below 450 and a log P below 4. And so that means that these compounds are particularly optimal starting point for further chemical decoration and optimization in SAR MedChem program. Here is a picture I wanted to show you of our I2PO screening station, in which we have integrated the two Tuckman machines. So by use of this I2PO screening station, we have a fully automated workflow, which starts from the cell lysate to the data released from the Tuckman machine. And the data which are released from this machine are directly automatically exported, so imported in our corporate database, which analyzes this data by the use of Syngenta Screener software. So now moving to a different classes of RNA tackling. So in this case is based on the interaction between protein and tRNA. So for developing such a kind of assay, we use different approach, because as you can imagine, all of them, they have pros and cons. So the first I'm going to show you is a TR thread base. So in this case, we have to tag our protein of interest and then to use a acceptor or donor level antibody. And then we use a tRNA, which of course has a specific sequence, which is recognized by the protein, which is biotinylated. And so in this case, we can then use streptavidin, which is labeled by donor or acceptor. Just to underline that when we develop this assay, we really try all the different combinations. And so different acceptor and donor and the different combination between the two entities in order to identify the most convenient combination, so the best performance assay and the most sensitive one. When the two players, so the protein and the RNA, they bind each other, so when we have the interaction, the donor and the acceptor, they come in close proximity, and so we have the emission of the TRFX signal. When we are looking for inhibitor of such a kind of interaction, we are expecting, of course, a strong reduction of the TRFX signal. So this is a sort of signal assay. And here I wanted to report you a case study for this kind of assay on which we run an high-throughput screening. And so these are the first step for the assay development and optimization, in which you see we have a dose increase of the tRNA in the presence of increasing concentration of the protein. And here you see that at the lower concentration, we have a very nice linearity of the reaction, and then all the conditions reach a plateau. Then after the identification of the optimal condition, we move to the assay validation. In this case, having no availability of reference compound, we use unlabeled, different unlabeled tRNA to validate that we were able to displace this interaction, so to interfere with this interaction. And so this assay is really suitable to identify the inhibitor of the interaction. Very important to underline is that we set up an interference assay in which only the readout component are used, while of course the protein and the tRNA are excluded from this combination, so that by using this interference assay, we can exclude all the compound which just interfere with the readout system. By screening 100,000 compound, in the end, we could identify 0.10% specific hit with a good affinity, so in the low micromolar range, and they were specific because, as said, all the false positive were excluded by the use of this interference assay. We can also use other approaches, so like the fluorescent polarization. So in this case, only the tRNA need to be labeled with the acceptor. Also in this case, the look for inhibitor implies the reduction of the fluorescence emission. Another possibility is the use of this alpha screen-based protein tRNA oligo interaction, in which, again, we need a protein tag and also a tRNA which is biotinylated. But in this case, instead of detecting fluorescence, we are detecting recording luminescence, which is activated, which is induced upon the binding of streptavidin donor beads to the complex, emission of singlet oxygen, which activate our anti-tag alpha screen acceptor beads. Also in this case, this is an off assay. To conclude this part on the interaction and between tRNA and protein, I also want to mention that in order to exclude from the very beginning potential liability, we also can use this fluorescent intercalator displacement method in order really to exclude compounds which are not really interfering with the interaction, but just intercalating the RNA, which of course is not a very good, I would say, outcome, because it can be very unselective and unspecific. Moving to the session of the messenger RNA expression assay, I wanted to report a couple of examples. So in this case, we use a phenotypic assay, and we wanted to follow the translocation of a transcription factor from the nuclei to the cytosol. Of course, the transcription factor is responsible for the expression of a specific gene. So we use an endogenous approach. And so in this case, we could follow the translocation by means of an antibody. And here you can see the result, in which upon the optimization of the assay by using different dilution of this antibody and the reference compound, which induce this translocation, we obtain a very nice result. But we also develop a parallel assay for the same target in a recombinant way. So in this case, we create a chimera between the transcription factor and a fluorescence tag, and we follow the translocation directly to the operetta without the need of any antibody. So here you can see the different conditions, so control condition in the presence of reference compound, plus a cofactor, and in the presence of reference compound and the cofactor. And here you clearly see that we have almost, I would say, no fluorescence in the nuclei, while a very strong fluorescence in the cytoplasm, and this corresponds to this curve. So the optimization of the assay in this condition allowed us to perform a screening, and so to identify compound able to induce this translocation. But for the specific messenger RNA expression, we can also use alternative approach. So Tachman-based assay, which is, I would say, similar to what I described for the alternative splicing. In this case, instead of focusing on alternative splicing, we would focus on specific gene expression by the use of the suitable probe. And we can also use a promoter reporter assay. And so in this case, what we do is that by use of CRISPR-Cas9 technology, we engineer the cell line and we insert a reporter gene, typically luciferase, at the 3' of the promoter. And so in this case, we can really follow the activation and the modulation of this promoter in a very native environment. And here I have an example of a cassette that we created, and also the result in which you see that we have a very high percentage of positive clone for what concern the insertion of the luciferase in the correct locus. And by the use of a reference inhibitor, we obtain a very nice dose response inhibition of the luciferase. So just to conclude, I wanted to report that we are also active in this developing assay and also following CIRNA treatment. So in this case, we use a Tachman-based approach. And so instead of incubated our assay plate, our cell plate with compound, we transfect the cell with specific construct which lead CIRNA. And so that we can detect silencing of a single gene or multiple gene, depending of the reaction. And here is just to show you that also in this case, we have to really set the condition by testing different parameters. And in this specific case, we had a very nice inhibition of our gene of interest in all the different conditions. When we tested a pool of CIRNA, we can use such a kind of assay to assess new targets. So to conclude, I would like to say that at Axxam, we can develop multiple assay types for drug discovery tackling RNA. And so these different assays are all developed in miniaturized format. We use a different readout system. We can use a Tachman-based screening by the advantage of this fully automated screening station that we have implemented. In all the cases, we generate interference and orthogonal assay to exclude all false positive hits, and we assemble a focused unique compound collection that we can give access to whom who are interested in. So this is the very final slide, and this is just to inform you that Axxam Bootcamp organized a next webinar, which will be held on the 16th of June, and which has as a target radiometric assays.
I want to thank both Stefan and Leah again for their presentation. If you have any extra questions to ask, you're welcome to send them to my email address and I will pass them on. And if not, thank you for joining us, and we hope to see you at the next webinar on radiometric assays on the 16th of June. So thank you very much.
Thank you very much.
Thank you.
Take care. Bye.
Bye.